Today I'm going to talking about endangered oaks, Quercus arbutifolia, in southern China, Montane cloud forest. So China is a huge country, and we have a very diversified ecosystems from the tropic, from subtropic, and also evergreen, and some deciduous, and also the high mountain area. So oakland is very important element in those ecosystems. And we also have a very rich legacy in the oak history, and as a timber, carbon, and also the framing food, and also tanning. And most important, its ecological functions, very important in those forestry ecosystem. So we have, in China, it's about 150 species oaks, and those oaks probably you guys never saw some of them, and it's very unique. And today we're going to talking about those one. This is leaves is a kind of rounded, and it's very rare. And here's the guideline. And okay, so. Arbutifolia actually is a kind of a shrubby looks trees, very shrubby, and usually the typical habitat is in the top of the mountain, very foggy, humid, windy, so mess up with the grassland. So this is the fruit, and it's like a biennial fruiting species, and usually we collect the acorns at usually the later of October, and we also can find the pistillate flowers right here. Where's it? Well, pistillate flowers right there. So it's definitely it's a biennial. And usually it's have a characters trade-off between the investment of a vegetative growth and also the fruiting. So at the year it's a kind of fruiting, it will not have any young branches growing. And when the young branches growing, you will not have the acorns. So usually the fruiting time at least needs 3 years to get the matured acorns. So that's the character. So look at the distribution of this oak. It's in the southern China and Indochina area. Here's the earth, and here's the only 6 distribution record of this oak. One, the type of material is coming from Vietnam. Sorry, I'm a little bit first. It's in the north of the Vietnam, and the other 5 are located in southern China area. Actually, it's a very huge area, but only those plants only distribute at the top of the mountain. This is the typical habitat of these plants. And the 2 on the left side are located in a nature reserve, and usually the population size of this one is the largest one. We call the Yizhang Manshan National Nature Park. And at the lower part, we can find it's messed up with mixed with Lizarcarpus hansii and also a bunch of other Castinopsis species. But at the top of the mountain, all the species, all these forests are composed by the Quercus arbutifolia, but the other 2 habitats is not that good. That's also in the CH habitats. It's in the national park also, but only have a very few individual at the top of the mountain. Another one, the ZZ population is also in some kind of tea garden or somewhere, and it's a lot of bamboo just surrounded. It's merged in the bamboo, the shrubby bamboos, and very few plants there, and very difficult to grow, have a difficult to reproduce themselves. So, and from 4 years the field survey, and we do trying to counting all the individuals of these endangered plants, and we found that's the distribution range of these 5 locality of these oaks. So most of them are located at the top of the mountain. This is the typical habitats. So okay, so we're going to talking about a little bit on the taxonomy at the very beginning, because I look at the Murphy's version. You send the list, then I noticed there are two names. The arbutifolia was listed. Actually, it was reduced to a phenomenon already. So Micropropagation was published and also recognized in Flora of China and also arbutifolia. But in the latest Flora of China English version, arbutifolia was reduced as a phenomenon. And when I doing my PhD work, I doing some little bit anatomy work, and I found the vena specimens, the leaves also rounded shape, and the trichons almost the same. And then I sampling the three of the species and compare with other groups, and we found some leaf characters. It's really interesting, and so it's demonstrated those three species are belonging to the same taxon. So it was reduced to phenomenon. So the two names are the phenomenons already. So the correct name should be Quercus arbutifolia. So that's. And so we'd like to know where the systematic position of the arbutifolia. So we construct the tree, thanks for Andrew, and we get the arbutifolia is right here. It's right here, and when we look at the image, zoom it in, and we found this clade is composed by biennial fruiting groups. So those groups are his very close allies. So, and we summarized our field collection and the field survey result, and we found some very interesting patterns, because the CH and ZY population usually they maintain a larger size of the population, and the individuals, the population structure seems to still have young plants growing and have a reproduction ability. But another three, I marked with the red. Those three have it seems have a reproductive difficulties already, because no seedlings was found under the ground. So probably, and also they seldom produce any acorns. So it's probably very critical situation already. So here I summarize the key characters of the Quercus arbutifolia. It's very rare, only three known locality, and it's a kind of inland nature, and all the habitats population are long distance isolated, and population size are usually very small, especially the three populations of less than 20 individuals was found. And the habitats are also very unique, because it's always located on top of the mountain, very humid, foggy, cool and windy area. And but it's also located on the biodiversity hotspot. And quite a few of the populations already show the sign of the difficulty on regeneration. So the aim of this study is trying to figure out what's the population genetic structure of the Quercus arbutifolia, and whether it's common comparing to the other common widespread lowland species or not. And another aspect is trying to figure out what's the restriction factors to the distribution of this species, and evaluate the threatening states of the Quercus arbutifolia on the background of climate change. And the last one is trying to apply the micropropagation technologies to rescue the population with regeneration difficulties. So here we're using the molecular markers to trying to figure out what's the population structure going to be. So we're using 4 chloroplast DNA sequence and plus nuclear DNA sequence ITS as the markers to do the approach. And we're using a bunch of cladistic softwares and also some population genetic software to trying to analyze the data. So we do get some result there. Based on the 4 DNA sequence, we get 22 variation sites and 16 haplotype. But when we look at the chloroplast DNA diversity, it's very high. HT actually is 0.9 And it's very rare for endangered species with a very small population already when we compare the result with the common one. And there are very significant differentiation between the populations because we can see the GST is much lower than the NST, the value. So it's indicated to have a very strong phylogeography signals. And it's also significant. IBD is indicate isolation by distance is very significant too. And we also can figure out there are some the population diversity level here, and those two populations pretty high, and those pines are the colors represent the haplotypes, so it's a very colorful one. It have more haplotypes within the population. For this one only have two colors, so only two haplotypes. So this one seems very suffer a lot from probably the bottleneck of fundus effects. So we did a little bit on the divergent time dating to trying to figure out what's the time of those haplotype diversified, and then we using 4 fossil as a calibration and figured out and that's the pre-quaternary diversification of the haplotypes. Those here's the Miocene, and the diversification ties begins with the later of Miocene right here, and the first lineage diversification seems happy at this stage and to the quaternary. So that's the main result. And let's back to the nuclear DNA. And from the ITS, we got actually ITS is a very messy result, and we have a lot of the haplotypes. And here we can see a bunch of colors, and those are the haplotypes in the populations. It's very colorful. It seems that ZY and ZZ population have the highest population level, diversification level, and also they show very high genetic diversity level because these values are also very high, and also very strong, significant phylogeography structure exists in the population. Then here's the network analysis, and it indicates that the population labeled with blue one, this one seems far away from the rest of the 4 populations. So it's a very unique population, although the haplotype is not that diversified. And the ITS is show the star-like pattern and probably suggested the recent range expansion. So we test a little bit on the SMOVA analysis. This test is trying to figure out whether there's a barrier between the populations. So when we saw the red dashed lines, the result from the ITS data, it seems that those 4 populations do not have a significant barrier between them. But the dashed line is the result getting from the chloroplast DNA. We can find, except those 2 clustered together, and the rest of the population seems to have a barrier between them. So that's the result. And we also can find both chloroplast and the nuclear DNA, the variation among the groups are greater, more than 50%, and the variation within population is greater than the 30%. It's very high already. And we did another analysis on the demographic range dynamic. Actually, the chloroplast DNA showed the structure like a kind of a hill. Those structures suggested that no population expansion was happened, but the ITS is a kind of smooth curves like that. It's suggested that probably the undergoing the population expansion, and we did a little bit estimation of the time of the expansion. It looks like at the later interligation of the quaternary. So another analysis is working on the extended bias screen plot analysis, and it suggested that the low and stable effective population size of arbutifolia begins since the beginning of the Quaternary, and it's possible that this species distribution range expanded, but the population growth is not happening. So this is the main findings of based on the population genetics. It's a high diversity of these rare plants, and here's the diversity level step down from those populations. And there are significant phylogeography structures, unlike other lowland Fagaceae trees. Usually the lowland Fagaceae trees like Quercus glauca, Quercus variabilis, their population only have a very few, almost no differentiation between the population. And the DM, the Damingshan population, is the early derived population and very isolated from the rest of the four. And the divergent tide, it seems, begin at the later of the Miocene, and is probably related to the formation of the Pearl River, because Pearl River probably is the main barrier between the one population to the other four population. So that's the main result we get. And here is another result study we did. It's on the morphology and the distribution. Based on the leaf morphological characters, we did some light microscope and SEM study on the trichome and stomata, and also analyzed trying to profile the habitat characters, so we get some result. First, here's the arm trichome. We found the three kinds of trichome, and SH is the Meihuashan population and Yizhang population. There's right here, they have less arms, and Yizhang is right there, and the arm is few, and the rest of the arms, trichome arms, is a kind of similar. So here's the trichome character. And we also get some result from the stomata density and the stomata area, and on the leaf lower surface of the trichome density, and also the upper leaf, the trichome density. And here, and friends help us to get the climate data for each site, and we do a little bit analyzing and figured out actually the 5 population, their climate factors are very different from each other, and we did some analysis using the R package of this one, and we can figure out the DM from the temperature parameters. DM and ZZ population, they have the highest parameters, and this one, SH, located have the lowest temperatures and the moisture. This one, moisture, the AP, the annual aspiration was found in CH, this one, CH. And Yizhang has the highest annual range of the temperature, so it seems the habitat is not exactly the same, very diversified. And when we look at the other characters, the leaf stomata, leaf area, comparing to the environmental factors, and we figured out that the leaf lower side of a leaf trichome density was negatively related to the stomata area, and the thermal parameters are negatively correlated with the stomata density, and the close relationship between the leaf lower side leaf trichome density and the stomata area to the annual aspiration and the EQ, the two parameter of the climate. So when we back to look at the habitats a little bit, and based on the future prediction, actually that prediction, that literature assessed the future climate change to 2050, and it's predicted that the mountain cloud forest will move upward. It's about 200 meters in the future, 50 years. So it's probably, look at those habitats. So those guys have nowhere to go, so probably will get extinct. And the only population have a good chance to survive is this one. This is the Yizhang population because they have elevation, it's about 200 meters and probably still can survive, maintain a very small population in this location. So the main findings based on the live anatomy and distribution is those habitats are very sensitive to the temperature and also moisture changes. And the live morphological plastic is happening, and it's a kind of a feedback to the warming and the drought climate change. And the cooling, humid are very essential to maintain big populations because all of the population with regeneration abilities are located in the north or the cooling area, so it's probably important for those species. And the southern populations, it seems, suffer a lot and very easy to get extinct in very recent few years. So how can we rescue those populations could not produce any acorns? So we're trying to figure out ways to trying to micropropagate them. So we did a little test on that. That's the 1 year seedling. Usually we put them into the growth chamber and keep very high humid, and then when we get the young branch, we use it as the explant, and here's the ways we prepare for the explant. So we're using this media to trying to get more shoots coming, and we optimize several concentration with the plant hormones, and we get the best combination should be this one for this species. Yeah. And we did a little bit, and after we get some shoots from the explant, then we cut it down and then transfer it to the rooting media, and then we get we test several combinations with the carbon, with different kinds of plant regulators, and then we get this one is the best one to work. So here's we succeed, but there's problems. We only succeed on the young plant, not the very old tree. Old trees are always very difficult to work on. So here's the rooting young plants in the tissue culture tubes. This is another one, Quercus arbutifolia. We also succeed in that one. So it seems working. And I'll try a little bit on the somatic embryogenesis with a live explant, but it seems 2,4-D is essential. We have to use it, but it's kind of when the culture on the dark culture should be much better than the light one. And we did some sectioning to observing which part of the parenchyma cell or callus to produce the embryo, and we thought it's probably the second callus generation. So it's just like it's kind of a global embryo or something, but we're still not very successful because it stopped growing, and we're still trying on that protocol. So the last part is the implication for the conservation. So the rare plants of arbutifolia still maintain very high population diversity, but think about it, it's very rare, and probably most of the population will gone, and it's better protect all of the population. So the south population suffer a lot from the degradation of the habitats and it's vulnerable to the global climate change. So should be have the priority to protect those. And because those Shanghai and Yijiang population located at the core area of the nature reserve, so in-situ conservation probably is good for those two. But most importantly, ex-situ conservation needs to be done for those three with the regrowth problems. So that's the result we get. So either we have to replanting them to the north cooler and humid area, or we need to stop cloning some of the, probably all the individuals we can get for this species. So that's all of it. And this was funded by National Science Foundation of China, Chinese Academy of Science, and a bunch of students and staffs working on the project. That's all. Thank you very much.